Hi, my name is Lauren Duvall, and today I'm going to be telling you a little bit about my research investigating morphometric differences of the cortex in individuals with autism spectrum disorder. Autism spectrum disorder, or ASD, is characterized by social impairments and restricted or repetitive patterns of behaviors or interests. My research project aims to understand how these symptoms might be linked to structural differences in the brain. The fusiform gyrus is located in the ventral occipitotemporal cortex, and it has different responsibilities in the left and right hemispheres of the brain. In the left hemisphere, it is responsible for visual word learning, and in the right hemisphere, it is responsible for face processing. Previous research studies have discovered differences in the neuroanatomical structure of the fusiform gyrus in individuals with ASD, but few studies have looked to see if these differences are related to aging and development or symptom presentation. My research study set out to see if development and/or symptom presentation, as represented by the Autism Quotient Questionnaire, could predict cortical thickness, surface area, and gray matter volume of the fusiform gyrus. We analyzed structural MRI data from 109 participants with an age range of 8 to 40. 51 of our participants had a diagnosis of ASD, and the other 58 were typically developing age and IQ matched peers. All of our participants completed the Autism Quotient Questionnaire, as well as the full scale IQ test, verbal IQ test, and performance IQ test. These scores will serve as our possible predictors. Then participants underwent a whole brain MRI scan. We analyzed our data with linear regression analyses. We ran our analyses independently in the typically developing and ASD groups and compared how well each of our possible predictors predicted a morphometric measurement of the fusiform gyrus. We found three significant results in our analyses. First, age proved to be a statistically significant predictor of cortical thickness and gray matter volume in both hemispheres of the fusiform gyrus in the typically developing group. This finding is consistent with previous research stating the trajectory of cortical thinning throughout childhood and adolescence. Since we did not find the same result in the ASD group, we proposed that could indicate abnormal cortical development. Our second major result suggested that autism quotient scores predicted the surface area of the right hemisphere in the typically developing group. Since the result was only apparent in the typically developing group and it was a negative prediction, we proposed that could be related to the face processing responsibilities of the right hemisphere fusiform gyrus. It is possible that the decreased surface area in the fusiform gyrus as a function of autism symptoms may underlie less practice and less developed face processing skills. Finally, full-scale IQ and verbal IQ were predictors of surface area in the left hemisphere of the ASD group, and verbal IQ was a predictor of gray matter volume in the left hemisphere of the ASD group. We propose that the significant negative relationship between verbal IQ and surface area and gray matter volume in ASD, especially in the left hemisphere, may reflect the role of the left fusiform gyrus in language processing and the associated deficits seen in ASD. Overall, the findings of this study provide important morphological differences in the fusiform gyrus in ASD, and that can explain the underlying face processing and social deficits in autism.